we looked at Fosgonimeton in models of inflammation. So we know inflammation is a key aspect of neurodegenerative disease, Alzheimer's, ALS, Parkinson's, contributes to ongoing neurodegeneration. And so we wanted to look at what Fosgonimeton could do in models of inflammation. We've seen before in some studies that we've seen anti-inflammatory activity. So we wanted to explore that a little bit. And so we presented a poster that was summarizing some findings of in vitro models with microglia. So microglia are the resident immune cells in the brain, and they can be triggered to go into this pro-inflammatory state. And we can do that in vitro. And when we did that, we use what's called LPS, which triggers the microglia to be activated, secrete all these pro-inflammatory cytokines like IL-1 beta, TNF alpha. But if we treat with Fosgo, we see a significant reduction in that pro-inflammatory state. We see a reduction in the expression of these inflammatory cytokines, which is really telling us that Fosgo can act on the microglia themselves and can modulate that immune response. And when we looked a little bit further into why that might be happening, what we presented was, Fosgo is modulating cell signaling within those microglia and actually reducing the production of reactive oxygen species. And it's the overproduction of reactive oxygen species by this microglia. They secrete these, and it damages nearby cells. And so if a treatment can reduce that, that would be beneficial to the neurons that would be around it. So that was an interesting set of data, and really indicates to us that Fosgo has this anti-inflammatory activity, and that could be important in neurodegenerative diseases such as Alzheimer's. And the second study that we presented, we had an oral talk at Society for Neuroscience, was looking further at Fosgonimeton in preclinical models of A-beta. So these are models of Alzheimer's disease where we use A-beta to drive, as the main challenge. And we found, we've done a lot of work previously that Fosgo was neuroprotective to various challenges, had not done a lot of work with A-beta or amyloid-β. So we did some of that work in vitro. We saw that Fosgo treatment could protect cultured neurons from amyloid-β-induced death. So really just protected them and made them survive even in the face of that pretty intense challenge. When you put amyloid-β onto neurons, it really does kill a lot of the cells. And so Fosgo was able to protect that. And one of the reasons that we think it was able to do that, it was protecting their mitochondria. And because amyloid-β can get into the cell, it can cause oxidative stress and disrupt mitochondrial function, which produces the energy for the cell. And Fosgo was able to prevent that disruption. And we think that's one of the main reasons why it was able to protect those cells. And so we saw that in vitro. And so then we looked at in vivo models of amyloid-β. So we presented some work in this mouse model where amyloid-β is injected directly into the hippocampus in the brain. So straight into the hippocampus, which we know is an area that is really critical to Alzheimer's. It's very important for normal cognitive function and memories. And so it's highly impacted in Alzheimer's. And so when you inject A-beta into that region, you see a lot of neuron loss, a lot of neurodegeneration, and then the animals exhibit cognitive dysfunction, cognitive impairment. And with Fosgo treatment, we were able to rescue that cognitive performance in several models or several outcome measures, such as the Y maze and the Morris-Water maze. Those are measures of spatial memory, and Fosgo was able to really improve the activity and the performance of those animals in those mazes, which would indicate a pro-cognitive activity. And then when we looked at the brains of the animals after the study, we saw that those that were given the amyloid-β injection, had a significant loss of neurons in that area where the injection occurred in the hippocampus. But those treated with Fosgo had a significant rescue, so much higher numbers of neurons in that area, which would suggest that Fosgo was able to prevent that neuron loss. So it was a good translation from our in vitro studies to this in vivo study where we saw neuroprotection against amyloid-β and saw reduced markers of apoptosis and things like that. So it was a really interesting study for us, a good translation from our in vitro. And I'll point out one more thing about this study was that we also looked at markers of neurogenesis. So we do know from the literature that this mechanism, which is targeting the neurotrophic HGF, can induce neurogenesis. That's been reported by others. And so we wanted to take an early look at that. It's not something we had done before. And we did see what we'll call increased cell proliferation in the subgranular zone. So that is an area of active adult neurogenesis. And so that gives us some indication that could be occurring. Now, we didn't do the additional staining to see if those neurons or those proliferating cells mature into neurons. So there's still more work to be done there. But it is an interesting and encouraging piece of data that we got from this study. So we're going to continue looking at that.